This is a brief overview of Chapter 5, the mitotic cell cycle. 5.1 Replication and division of nuclei and cells. 1. Describe the structure of a chromosome. Let me show this to you. Hold on. So at the end caps here, you have the telomeres, and those are just, we'll get into that more in a minute, but those are just areas that have no important DNA and protect the genes during replication. And then you have the centromere, which is this whole area right here connecting the sister chromatids, which are, this is a chromatid, and both of these together are a sister chromatid, and then this whole thing is the chromosome. And then each chromatid has its own kinetochore. So there are 2 kinetochores per chromosome. And then the chromosome is part of, so you have your DNA, and that wraps around the histones. And then you have a nucleosome, and this is wrapping around, and it's coiling into chromatin, and then it's supercoiling within the chromosome, and then you have your chromosome. So DNA stores the genetic information of the cell. It is made up of deoxyribose, which is a sugar, phosphate, and a nitrogenous base, either adenine, thymine, cytosine, or guanine. DNA wraps around a histone about 2 1/4 times. Histone proteins help to package DNA into a smaller space, so it provides surface area for the DNA to wrap around so that it doesn't knot. Not as in KNOT. Sister chromatids are replicated DNA. A pair of sister chromatids form a chromosome. Centromere is the area where the sister chromatids attach. It is the chromosome locus responsible for the segregation of genetic material. There are no genes located at the centromere. Telomeres are end caps on the chromosome. They contain no important DNA and keep information from being lost during DNA replication. 2. Explain the importance of mitosis and the production of genetically identical daughter cells during growth of multicellular organisms. All living organisms grow and reproduce. Living organisms are made of cells. Cells must be able to grow and reproduce. Cells reproduce by dividing. In diploid cells, this is mitosis. Diploid cells being, in a human and an animal, it's everything except your gametes, which are sex cells, and passing on copies of their genes to daughter cells. This process must be very precisely controlled so that no vital genetic information is lost. The 2 daughter cells formed after mitosis have the same number of chromosomes as the parent cell and are genetically identical. This allows growth of multicellular organisms from unicellular zygotes. Replacement of damaged or dead cells. This is possible using mitosis followed by cell division. Cells are constantly dying and being replaced by identical cells. The chromosome number of each of the offspring nuclei is the same as the parent nucleus, and the genome is kept exactly the same. Mitosis maintains the same chromosome number and genotype throughout life, growth, and repair. Mitosis ensures that every body cell throughout life, except gametes, has a genome identical to that of the original zygote. Repair of tissues by cell replacement. Mitosis creates cells identical to the original. In tissue repair, this ensures that the new cells function the same as the dead cells. Asexual reproduction. Asexual reproduction is the production of new individuals of a species by a single parent organism. The offspring are genetically identical, a feature that is only able to occur due to the production of genetically identical daughter cells in mitosis. So there's 2 ways that you might see asexual reproduction of an entire organism. So with a single-celled organism, an amoeba, just undergoing mitosis is obviously going to create a new organism. But with plants, they can also asexually reproduce, and that involves budding off, which is kind of when part of the plant comes off and then goes on to another plant. Kind of makes its own little guy. 3. Outline the mitotic cell cycle. So the mitotic cell cycle has 3 stages: interphase, which is G1, S, and G2, and then mitosis or nuclear division, and then cytokinesis. And then these little stars right here are checkpoints. So at the end of G1, G2, and mitosis, the cell undergoes a check to make sure that it's growing properly. And if not, the cell will undergo apoptosis, which is programmed cell death. So interphase has 3 stages. G1, the cell grows to optimum size. So with the G stages, the G stands for gap, but you can think of it as growth if that helps you remember what it does. Protein synthesis occurs to make proteins involved in synthesizing organelles. Cell is checked that it is the correct size, has the correct nutrients, growth factors, and that there is no damaged DNA. If the cell doesn't pass these checks, replication will not continue. S phase. This is synthesis. The S in S phase stands for synthesis. DNA is replicated, so the DNA is synthesized. And then in G2, the organelles and proteins are made. The cell continues to grow, energy stores increase, and the newly replicated DNA is checked for copying errors. So, in summary, G1, the cell grows to optimum size. That's very important. It's very important that you use the word optimum. But also, just know that protein synthesis occurs to make the proteins needed to replicate organelles, and that there's a checkpoint. And then S phase, the DNA is synthesized. And then in G2, there's the organelles are replicated, proteins are made, and there is another checkpoint, as well as cell growth is occurring. And also, it's important. Oh, I'm getting there. Why is resting stage not an appropriate description of interphase? So, during interphase, the cells are metabolically active, protein synthesis is occurring, transcription and translation is occurring, gene expression is happening, DNA replication is occurring, and the organelles are synthesizing. And for all those reasons, you can't say that interphase is a resting stage, because the cell is not resting at all. That cell is not asleep. That cell is very active. Okay. Mitosis, or nuclear division, has 4 stages. The first is prophase. So, during prophase, chromatin condenses into chromosomes and the nucleolus disappears. The chromosomes condense and become visible. In animal cells, the centrioles separate. The centrioles move to opposite poles of the cell. The centrioles are responsible for creating spindle fibers. Spindle fibers are released from both poles to create a spindle apparatus used in later stages. Plant cells have a spindle apparatus but lack the centrioles. And then over here, this is a very simple diagram of prophase, and you also need to know how to identify it in a photomicrograph. So make sure you familiarize yourself with that. I can't help you there because my printer doesn't work. So during early prophase, the nucleolus is still present, the nuclear envelope is still present, and the chromatin is just a chromatin network. It's just kind of everywhere. But in late prophase, that chromatin has condensed into chromosomes, or chromatids, which have paired together into chromosomes. And the nuclear envelope is still present. Stage 2 is metaphase. The chromosomes line up along the metaphase or equatorial plate. It's important that you either say metaphase plate or equatorial plate, because if you just say the middle, you're wrong. So the chromosomes align along the equator of the cell. The spindle fibers are released from the poles. The spindle fibers released from the poles now attach to the centromere and chromatid. And then again, another little simple drawing. You have the centriole at each pole, the spindle attaching to the chromosomes at the equator, and the nuclear envelope has disintegrated. Now, so you might see that phrase, the nuclear envelope has disintegrated. That's not entirely true. It hasn't disappeared. It's not gone. It's just broken down into small vesicles that can no longer be seen under a microscope. So it's easier for textbooks to just say that it disappeared. But in case you get asked about it, just remember it's not gone. It's just too small to see with a microscope now because it's formed vesicles. Okay, stage 3 is anaphase. The chromosomes break at centromeres and sister chromatids move to opposite ends of the cell. The spindle fibers start to retract and pull the centromere and chromatids they are bound to towards the opposite poles. This causes the centromere to divide in two, and the individual chromatids are pulled to each opposite pole. These separated chromosomes are now referred to as chromatids. This stage requires energy in the form of ATP, which is provided by respiration in the mitochondria. So, in case it asks you why this might require ATP, I imagine you know this, but just think about the fact that these spindle fibers are having to actively pull the chromatids away from each other and toward the poles. So because that takes energy, that needs ATP. So anaphase is relatively simple. You just have your chromatids in this like V shape. It's pretty important, and it's important to show that the spindle fibers are attached to the kinetochore. The final stage of mitosis is telophase. The nuclear membrane reforms, nucleoli reappear, and the chromosomes unwind into chromatin. The chromosomes are now at each pole of the cell and become longer and thinner again. The spindle fibers disintegrate and the nucleolus starts to reform. So during this stage, the chromatid is starting to unwind, but I drew it like this. You're allowed to draw it like this. I drew it like this just to show that the sister chromatids have gone to either new cell. So this is gonna be like daughter cell 1, that'll be daughter cell 2, and then this one is like that sister chromatids with this one, that one's sister chromatids with this one, and this one is sister chromatids with that one. That's not like something you'll have to specify, but it's just important to remember that it's not like they're each getting one copy of the genes of the DNA. The final stage of the cell cycle is cytokinesis. It's the final stage of cell division when the cytoplasm divides to form two separate daughter cells, which you can see here, and the chromosomes is unwind. Oh, there should be a nucleolus there. The little guy. In animals, a cleavage furrow develops and runs around around the cell in a line with the original equator of the spindle. Once daughter nuclei have formed, the cleavage furrow deepens and eventually cuts the cell in two. In plants, phragmoplast or cell plate is formed between the two daughter nuclei. This cell plate is assembled from membranous vesicles from the Golgi body and endoplasmic reticulum. The vesicles give rise to the middle lamella of calcium and magnesium pectates on which cellulose is deposited. And then all in all, the two new genetically identical cells then pass into the G1 phase of the cell cycle. 4. Outline the role of telomeres in preventing the loss of genes from the ends of chromosomes during DNA replication. The main function of telomeres is to ensure that the ends of the molecule are included in the replication and not left out when DNA is replicated. Telomeres work by making DNA a bit longer. They have no useful information, but allow the copying enzyme to complete copying all the needed DNA. The enzyme used to make telomeres is telomerase. This enzyme basically just makes the telomeres longer, or adds to the telomeres so that no DNA is lost no genes are lost from the DNA. The main function of telomeres, therefore, is to prevent the loss of genes during cell division and allow replication. Some cells do not top up their telomeres at cell division. Therefore, with each division, the DNA gets smaller and smaller until vital DNA is no longer protected. So the cell dies, then the organism ages, then the organism dies. Telomeres shorten with age, but this does not begin occurring until after you have been birthed. 5. Outline the role of stem cells in cell replacement and tissue repair by mitosis. A stem cell is a cell that can divide an unlimited number of times by mitosis. Each new cell has the potential to remain a stem cell or differentiate into a specialized cell. The power of a stem cell to produce different types of cells is variable and is referred to as its potency. Stem cells that can produce any type of cell are called totipotent. For example, zygotes up to the 16 cell stage of development. So as soon as the sperm fertilizes the egg, this zygote is totipotent. So it can produce any type of cell that it needs, that it could possibly need, and then it forms the blastocyst. It becomes a blastocyst, there is a placenta, and now it's not totipotent anymore, it's pluripotent, which means it can make a lot of cells, but it can't make every kind of cell. So embryonic stem cells are pluripotent. For growth and repair, some stem cells remain multipotent. For example, stem cells in bone marrow produce red blood cells, monocytes, which are white blood cells, neutrophils, and lymphocytes. So those are multipotent stem cells. So adult stem cells, your stem cells are just multipotent. So totipotent stem cells can divide and produce any type of body cell. During development, totipotent cells translate only part of their DNA, resulting in cell specialization. Okay, so cell specialization, that's important. So you have to use the word differentiate when you're talking about specialization, so they differentiate into another type of cell. So they have all the same genes. It's not like so they have all the genes. It's just when they form this other cell, they turn on the genes that are necessary for that cell's functioning. So they differentiate into a specialized cell by activating the genes necessary for that type of cell's functioning. Totipotent cells occur only for a limited time in early mammalian embryos. Pluripotent cells are found in embryos and can become almost any type of cell. For this reason, they are used in research with the prospect of using them to treat human disorders. There are issues with this, as sometimes the treatment doesn't work or the stem cells continually divide to create tumors. Multipotent cells are found in mature mammals and can divide to form a limited number of different cell types. You might be asked about the ethical debate of using stem cells for research. It pretty much boils down to for it to be useful for research, it has to be an embryonic stem cell. So you have to take an embryo and just use those stem cells for research, and then the debate there is you could make that embryo into a person, or that could have been a person. So that's the debate there. Sorry, my rabbit's on the floor. Hi, baby. So yeah, the debate there is just that the embryo could have become a person, but instead you're using it for research. There's also the debate with producing new organs for someone, like if they need an organ transplant, and so the decision's made to use their stem cells to create that organ so that their body doesn't reject it. They're creating this clone embryo of them. They're creating this clone embryo of them and then destroying it once they have their stem cells. So again, there's this debate of that could have been a person. So yeah. 6. Explain how uncontrolled cell division can result in formation of a tumor. Mitosis is a gene-controlled process, but mutations in genes that control when cells divide can result in uncontrollable mitosis. Uncontrolled cell division can lead to the formation of tumors and cancer. Malignant tumors are cancerous because they grow rapidly and can spread to other tissues in the body and often have their own blood supply. Cancers start when changes occur in genes that cause cell division. A change in any gene is called a mutation. The term for a mutated gene that causes cancer is called an oncogene, which is ONCO gene. So the steps for like an oncogene to become a tumor. So step 1, oncogenes are transformed by carcinogens, which carcinogens are just like anything in the environment that are cancerous, so like asbestos. Step 2, cancerous cells do not respond to the signals from other cells, so it continues to divide, which is just saying like those checkpoints we talked about earlier, when those alarm bells go off and it's like, wait, you're dividing too fast, this is happening all wrong. I'm like, no thank you, I'm gonna keep doing my thing. And then step 3, mitosis occurs, and then because of that because mitosis is occurring, that mutation in the genes is transferring to another cell. So step 4, the cancerous cells are not removed by the immune system and still absorb nutrients. So that one's pretty explanatory. They're not removed from the immune system, and they're absorbing your body's nutrients. So they're not adding anything to your body. They're just taking away nutrients from your other cells. Step 5, rapid mitosis occurs. So that's that like. Step 6, the tumor gets bigger because of said rapid mitosis. The cell change their characteristics and look different under the microscope. That is likely also because of the rapid mitosis, because they're going too fast. They're not exactly going through mitosis the way they should. So they're just kind of dividing whenever they want without finishing mitosis. So that creates weird shapes. Little funky guys. Little weirdos. And then step 7, the tumor is supplied with blood and lymph vessels. Tumor cells spread in blood and lymph to other parts of the body. And then this is step 8. That's metastasis or metastasis. It doesn't matter how you say it as long as you spell it right. The tumor cells invade other tissues, secondary cancer throughout the body. So you have your tumor, and then because it's being supplied with that blood and lymph, part of it breaks off and goes into the bloodstream or the lymph, and then it spreads to another part of your body, and then you have the secondary tumor, and then that's growing too, and so the cancer's spreading. Okay, so that is all for replication and division of nuclei in cells for Chapter 5, the mitotic cell cycle. Good luck. Study. You will do fine. I love you.